Uh, Moving on to culture media, continuing CBMA lectures. Uh, Continuing the CBMA lectures, moving to culture media. What is culture? Culture is one of the essential things. It's one of the essential things in Robert Cox postulates. The organism should be constantly associated and should be cultivated with pure culture in the laboratory. Culturing the organism is growing the organism, media on which we grow the organisms. Culture media is very important for microbiology exam going students because they are kept as spotters. They will be asked in oral examination and they can come as a question in the theory exams also. So you should be thorough with culture media. Students neglect this, seeing that some colorful plates and some reaction in the test tubes, black and white color and all those things. So please remember, culture medias are very important from exam going student, both for practicals, viva and theory exams. So culture is classified depending on the consistency as solid, liquid and semi-solid. Liquid media, all the organisms look alike and all the organisms look correct. They form turbidity or pellicle on the surface of the medium. Solid media is converted to liquid media is converted to solid media to identify the colonial characters of the microorganisms. You can distinguish one organism from another organism. And liquid media is converted into solid media by two solidifying agents. One is gelatin and another is agar-agar. Agar-agar is a seaweed. Gelatin is used by some of the organisms like anthrax and clostridium, and it will be set at refrigerated temperature. It is very costly. 15% of gelatin is needed for preparation of liquid into solid media. So gelatin is not commonly used for preparation of solid media. The most commonly used is agar powder. You need 2% of agar powder. It melts at 96 degrees and sets at room temperature. So agar is inert substance, is not utilized by, it has no nutritional value and it is used for preparation of solid media. And then semi-solid media, when they put dispense 0.5% of the agar, it is called semi-solid media. And semi-solid media is example of mannitol motility media. It's commonly used for to test the motility of the organisms. Now, depending on the use, they are classified into simple media, enriched media, enrichment media, differential media, selective media, anaerobic media, media for biochemical reactions. So you should remember, it's very easy if you memorize and you can identify the organisms. So most commonly used peptone water and nutrient broth. Now moving on to the peptone water, the nutrient broth and peptone water are simple media. Complex media is to bring out the characteristic of the microorganism. Synthetic media is used for growing the organism with minimal requirement. Enrichment media, when you add blood, ascitic fluid, serum, it becomes enriched media. Enrichment media is used, it's our liquid medias, example selenite broth and tetraethanate broth. It is used when pathogen is mixed with commensal. For example, if you want to isolate Salmonella and Shigella from stool, feces, enrichment, please remember, are liquid media which enhances the growth of pathogens and inhibits the growth of commensal. Enhances pathogens, inhibits commensal. Enhances pathogen, inhibits commensals. Example is tetrathianate broth and selenite broth. So blood agar is most commonly asked as a water or in the examination, and it is you have to identify that it's red in color, and a lot of people confuse it with McConkey agar, which I'll tell you in detail. Blood agar is a enriched media. 7% of sheep RBCs are added to nutrient agar, and blood agar is enriched media as well as differential media. Differential media, it differentiates streptococci into alpha, beta and gamma streptococci. Alpha is partial zone of hemolysis, beta hemolysis is complete zone of hemolysis, and gamma is non-hemolytic. Alpha Streptococcus virilis, example Streptococcus virilis. Beta hemolytic streptococci, again they are classified to A to U by Rebecca Lansfield, and A is pathogenic, further classified by Griffith 95 types. Gamma is enterococci or Streptococcus faecalis, normal commensal in the intestine, but is one of the important causes of urinary tract infection. Now, blood agar, sheep blood is used, and again I said it's differentiate between alpha, beta and gamma hemolytic streptococci. And chocolate agar is heated blood agar, and it is heated for 70 degrees. It hits 70 degrees for 25 to 30 minutes. It becomes chocolate agar, and chocolate agar is useful for growing hemophilus organisms that are H. influenzae, meningococci and gonococci, and it is also useful for growing pneumococcus. And the advantage of chocolate agar or blood agar, RBCs are broken down, the hemoglobin is released, and the organisms are, it's like peeled banana. So these organisms can utilize the nutrients easily and grow them, and it has got X factor, that is hemin, and V factor, NAD, which are X and V factor, we call it as for growth of hemophilus influenzae. Brain heart infusion broth is used for collection of the blood for culturing, and it is nutritious, and you can grow the fastidious organism in it. And enrichment media, as said, tetrathianate selenite broth. Selective media is used for inhibiting unwanted organism and enhancing the pathogen. For example, Wilson and Blair's bismuth sulfate agar. It is used for cultivation of Salmonella in the stool. Desoxycholate agar, again the same thing, for the use of growing the Salmonella from the TCBS, tharocolate citrate sucrose agar, for growing the cholera bacilli. And Lowenstein junction media, it is an egg medium. It contains malachite green, mineral salts, and hen's egg, which is sterilized by intermittent sterilization, partial sterilization, or tindelization. And generation time is defined as time taken for the one binary fission of the bacteria. It is 20 minutes in E. coli, 20 minutes in E. coli, 20 hours in tuberculosis, 20 hours in case of Mycobacterium leprae. Remember 2020, IPL 2020 or 2020 matches, World Cup. 20 minutes E. coli, all pathogens, 20 hours for tuberculosis, 20 days for Mycobacterium leprae. So Mycobacterium tuberculosis gives rise to white mat colonies, rough, buff, tough, non-tenacious colonies on the Lowenstein junction medium. So TCBS for cholera, Wilson and Blair for isolation of salmonella from stool, LJ for tuberculosis. And potassium tellurite agar, when you add potassium tellurite to the chocolate agar, it becomes potassium tellurite. Tellurite is reduced to tellurium by diphtheria bacilli. Colony looks black in color. So now culture media, differential media, one of the important that is most commonly asked in your examination is McConkey's media. Please remember the mnemonic plant, PLANT. Peptone, lactose, agar, neutral red, and tarocolate. Tarocolate is a selective agent which inhibits non-enteric organisms. So it differentiates between lactose fermenters and non-lactose fermenters. Lactose fermenters are pink colored colonies, non-lactose fermenters pale and dark. Example of lactose fermenters are E. coli and Klebsiella. Non-lactose fermenters are Amila, Shigella, Proteus, and Pseudomonas. Remember EK for lactose fermenter, E. coli and Klebsiella. Non-lactose fermenters SS, Salmonella, Shigella, Proteus, Pseudomonas. SSPP. Please remember this. Plant mnemonic for the McConkey's medium. And that's a differential media for the some of the transport media that are used for fastidious organisms, for the delicate organisms. Stewart media for cocci, Carey Blake's medium for enteric bacilli, buffered glycerol for enteric bacilli. Now anaerobic media is to reduce the oxygen, and example is liquid medium is thioglycolate. Robertson Cook medium, minced meat is added to absorb the oxygen in the medium. So remember this: Robertson Cook media and thioglycolate media. So you should remember they are poured in the petri dishes. Petri dishes got the name, it was discovered by Petri, and this is how in a nutshell we remember about culture media.